Imagine, for a second, that you're looking at a lab report for a drug designed to target a super aggressive cancer.
Right, a highly controlled scenario.
Exactly. And the data shows that when you combine two specific chemical compounds in a perfect 1-to-1 ratio, the results are just miraculous.
Yeah, like it aggressively induces cell death in the tumor.
Right. It literally physically stops the cancer from building the blood vessels it needs to survive.
Yeah.
You think, wow, we found a massive breakthrough.
But then you run the experiment again.
Yeah, same exact compounds, same exact cancer cells. The only thing you change is the ratio. You shift it to, like, 1 part of the first compound and 4 parts of the second.
And suddenly the drug doesn't just stop working.
No, it actively hinders the treatment. It creates this entirely antagonistic environment that could, theoretically, allow the cancer to spread faster.
It is honestly one of the most terrifying and, well, deeply fascinating paradoxes in modern botanical pharmacology.
It's wild, right?
Yeah. The exact same ingredients yielding wildly opposing biological consequences, dictated entirely by these tiny microscopic shifts in their mathematical ratio.
And, if you're listening to this right now, that exact paradox is the reason we are doing this deep dive tailored specifically for you today.
Because you want to understand what is actually happening in the body, without all the noise.
Exactly. Because arguably no topic in modern medicine is surrounded by more marketing hype, more anecdotal muddy waters, and honestly, more political baggage than the science of cannabis.
Yeah, and we have an absolutely massive stack of literature to go through today. The pile on the desk is literally intimidating.
Oh, for sure. We're looking at comprehensive pharmacological reviews from the journal MDPI, right?
Yep, plus extensive clinical trial data presented to UK Parliament committees.
And we've got neurobiological breakdowns from Frontiers in Pharmacology and clinical surveys from the Beth Israel Deaconess Medical Center.
It's a lot, but our mission today is singular.
Right. We are going to completely strip away all that colorful packaging, ignore the dispensary marketing claims, and just look strictly at the rigorous peer-reviewed science.
We're basically going to follow the molecules.
Yeah. First, by looking at the biological hardware, like what your brain and your cells are actually doing at a molecular level when these compounds enter your system.
Which includes stuff like neuron firing patterns, memory consolidation, and synaptic pruning.
We'll definitely get into that pruning stuff because it's fascinating.
Yeah.
Then we'll look at the psychological reality.
Right, what the patient actually feels.
Yeah, how microscopic aromatic oils can physically dictate whether you feel a wave of anxiety or just like profound sedation. Why your dreams feel real, or why poor sleep equals brain fog.
And an emotional reset during REM sleep.
Exactly.
Yeah.
And finally, we will look at the behavioral outcomes, the real-life impacts on eating, sleeping, how focus drops, or reaction time slows.
And how patients are actively using this chemistry to manage some really grueling clinical symptoms.
Because, I mean, it's the only logical way to untangle this subject, right?
Oh, absolutely. You simply cannot understand the behavioral changes, like why someone's focus suddenly drops, or why their creativity is stifled without understanding the cellular biology that caused it.
So, let's start at the very beginning. Before we can even begin to talk about how this plant affects the human brain and body, we sort of have to look in the mirror.
Right, we have to understand the biological hardware that actually receives it.
Because honestly, why does the human body even have a reaction to a random botanical shrub?
Right, it's not like we have specialized receptors just waiting around on the off chance we ingest, like a fern or a maple leaf.
Exactly.
Yeah.
So what's the deal?
Well, the answer to that fundamentally changed neurobiology. And what is wild is that it wasn't even discovered until the early 1990s.
The 90s. That is practically yesterday in the grand scheme of medical science.
I know, we were sequencing the human genome, but we had absolutely no idea about this massive cellular network.
You're talking about the endocannabinoid system, right?
Yeah.
The ECS.
Exactly. Some molecular biologists actually refer to it as the endocannabinoidome.
Because it's just everywhere.
Yeah, simply because its reach across human physiology is so incredibly vast.
Okay, so how did they actually find it in the 90s?
Well, in the late 80s and early 90s, researchers were essentially trying to reverse engineer a drug high.
Okay, that makes sense.
Yeah, they wanted to figure out exactly how THC, which is the primary psychoactive compound in cannabis, was binding to the brain.
They were looking for the lock that this plant key fit into.
Exactly. And in doing so, they stumbled upon an entirely unknown deeply complex cellular signaling network that already existed inside all of us.
And it wasn't there just for the plant, obviously.
No, not at all. The ECS is essentially the human body's master regulatory system. Its sole biological mandate is homeostasis.
So basically, it's an internal thermostat.
That is a perfect way to visualize it. Think about the systems in your house. If the ambient temperature gets too high, the thermostat registers that deviation and kicks on the air conditioning, right? It cools it down to a baseline.
Right.
So if your biological stress levels spike, or if your neuronal firing is just out of control, the ECS kicks in.
Like if your immune system is overreacting to an injury and causing massive inflammation.
Exactly. The ECS steps in to bring your cellular environment back to a stable baseline.
So how does it physically do that? Like, what are the mechanisms of this biological thermostat?
Well, the literature keeps pointing to two primary pieces of hardware, the CB1 and CB2 receptors.
Okay, let's break those down.
Those are the two most heavily studied receptors in the system. And their physical locations in the body pretty much dictate everything about how they behave.
We're the CB1 receptors.
They are highly abundant in the central nervous system. We are talking about massive concentrations in the brain and the spinal cord.
So they're dealing with the cognitive stuff.
Right. They heavily influence memory consolidation in the hippocampus, motor control in the basal ganglia, and your pain perception pathways.
Okay, and the CB2 receptors, where are those?
CB2 receptors are an entirely different beast. They are predominantly restricted to immune tissues and cells.
Like what?
Things like your spleen, your tonsils, and the leukocytes traveling through your bloodstream.
Oh, wow. So they aren't really in the brain at all.
Not primarily, no. Their primary function is modulating inflammation and the immune response.
Okay, but this brings up a massive biological question.
Which is?
Well, if we had these CB1 and CB2 receptors just sitting in our brain and our immune system, the body must produce its own internal chemicals to bind to them, right?
Oh, absolutely. The system had to exist long before humans ever interacted with the cannabis plant.
Right, endo meaning internal.
Yes. The discovery of the receptors immediately launched a massive hunt for the body's natural internal cannabinoids. And we found them.
What are they?
The two primary endocannabinoids are anandamide, which actually takes its name from the Sanskrit word for bliss.
Oh, so it's the bliss molecule.
Exactly, it's often called that. And then there's a second compound called 2-AG.
Anandamide and 2-AG. So how do they actually work in the brain? Are they just standard neurotransmitters like, dopamine or serotonin?
No, and this is where the biology gets incredibly elegant. Most standard neurotransmitters are synthesized ahead of time.
Like they're stockpiled.
Right. They're stored up in little biological bubbles called vesicles at the end of a neuron, and they just sit there waiting for an electrical signal to trigger their release across the synapse.
The synapse being the gap between the neurons.
Exactly. They cross the gap to pass the message forward. But endocannabinoids break that rule entirely.
Right. How?
They are synthesized on demand, directly from the lipid membrane of the cell, only when they are actually needed.
Wait, really? They're made out of the cell's own structural fat.
Yes. Let's paint a picture. Imagine a neuron is being bombarded with excitatory signals because you're experiencing physical pain or severe psychological stress.
Okay, so the neuron is firing rapidly.
Right, it is essentially screaming. This overstimulation opens calcium channels, which acts as a trigger.
And that tells the cell to make the bliss molecule.
Precisely. The screaming neuron instantly synthesizes an anandamide or 2-AG from its own membrane. And here is the real kicker. Instead of traveling forward, these endocannabinoids travel backward across the synapse.
They go the wrong way.
Yeah, they engage in what is called retrograde signaling. They travel upstream.
That is so weird. Why do they go backwards?
Because they cross the gap backwards to find the CB1 receptor on the neighboring neuron, the one that is sending all those screaming excitatory signals.
Oh, I see.
They bind to the lock and basically tell that sending neuron to shut up. They actively dial down the release of the excitatory neurotransmitters.
It's a biological read receipt.
A read receipt.
Yeah, like when someone is sending you 30 frantic text messages in a row and your phone sends back a signal saying, message read.
Oh, that is a brilliant analogy.
It tells the sender they can stop firing. The message got through.
Yes, exactly. It stops the cascade of panic. And once that message is received and the cellular balance is restored, the body immediately cleans up the mess.
How's it clean it up?
Specific enzymes rush in to break the endocannabinoids down. An enzyme called FAAH breaks down anandamide, and an enzyme called MGL breaks down 2-AG.
So the messages are destroyed almost as quickly as they are created.
Exactly. It is a highly regulated, tightly controlled biological loop.
But looking at the pharmacological reviews here,
Yeah.
they point out something really critical. These cannabinoids, both the ones our bodies make and the ones from the plant, they don't exclusively talk to CB1 and CB2 receptors, do they?
Not at all. The thermostat is wired into a lot of different systems. The biological reality is honestly staggering.
So what else do they hit?
The pharmacological actions of cannabinoids target an extensive, complex array of receptors across the entire body. For instance, they bind to TRPV1.
TRPV1, what does that do?
It is a receptor heavily involved in sensing pain and regulating your body temperature. Fun fact, it's actually the exact same receptor that gets triggered when you eat a painfully hot chili pepper.
Oh, wow. So the body uses endocannabinoids to modulate that pain response.
Yep. They also target GPR55, which is an orphan receptor involved in cell proliferation, and crucially, they target the 5-HT1A receptor.
5-HT1A. Whenever I see 5-HT in the literature, I immediately think of serotonin.
And you're spot on. It is a vital part of the serotonergic pathway.
Okay, so mood regulation.
Exactly. It's a G protein coupled receptor heavily involved in regulating your mood, mitigating anxiety, and altering pain perception.
So it's not just a single light switch.
No, it is the master control board for a massive physiological network, which is why the psychological and behavioral impacts of the ECS are so profound.
Okay, so I want to step into the shoes of the learner listening to this for a second.
Okay, go for it.
We have this beautiful, tightly regulated internal system. We make our own bliss molecules on demand. They travel backward to quiet down screaming neurons. Enzymes clean them up.
Right.
It sounds totally flawless.
Yeah.
So why does introducing plant-based cannabinoids cause such dramatic biological shifts? I mean, if the internal thermostat works, why do we need a plant at all?
That is basically the fundamental question of cannabinoid medicine. The reality is that the human body is subjected to intense wear and tear.
Like from stress.
Disease, severe trauma, chronic stress, or long-term neurodegeneration. All of that can cause that internal thermostat to fundamentally break down.
Oh, so the baseline gets broken.
Precisely.
Yeah.
In conditions of severe chronic pain or systemic immune dysfunction, the body's natural on-demand production of endocannabinoids simply might not be enough to restore homeostasis.
It just gets overwhelmed.
Yeah. Or those cleanup enzymes, the FAAH enzymes, might be too aggressive. They might break down the anandamide before it can even do its job.
Right.
Or the receptors themselves might become desensitized. We actually call this clinical endocannabinoid deficiency.
Okay, and this is where the botanical chemistry steps in.
Exactly. The ECS provides the biological locks, and our internal endocannabinoids are the natural, native keys.
But the cannabis plant has its own keys.
Right. Through millions of years of its own botanical evolution, the plant produces over 100 different chemical keys that miraculously fit into those exact same human locks.
That's crazy.
Sometimes they perfectly mimic our natural messengers, sometimes they block them entirely, and sometimes they physically change the shape of the lock itself.
Well, let's follow that transition, moving from the human body over to the botanical chemistry. Let's look at the actual chemicals trying to unlock these doors.
The major and minor phytocannabinoids,
Right, phytocannabinoids meaning quite literally cannabinoids derived from a plant.
as opposed to endocannabinoids derived from inside the body, yes.
And obviously everyone knows the two lead actors on this stage, THC and CBD. But biologically, when they enter the brain, they play entirely different roles, don't they?
Oh, their pharmacological mechanisms could not be more different. Let's look at THC, or delta-9 tetrahydrocannabinol.
Okay.
THC is what pharmacologists call a partial agonist of the CB1 and CB2 receptors,
Meaning what exactly for the listener?
meaning it binds directly to the primary active site of the CB1 receptors in the central nervous system. It acts as a direct mimic.
So it looks like our internal keys.
Yeah, THC looks enough like our natural anandamide that the receptor accepts it, and that direct binding in the brain is what causes the classic psychoactive effect, the high.
So it essentially hijacks the retrograde signaling system.
It does, but with a massive caveat. Remember those cleanup enzymes we talked about?
Yeah.
FAAH.
The ones that destroy our natural endocannabinoids once the message is delivered.
Right. Well, FAH doesn't work on THC.
Clearly, it doesn't break it down.
The enzyme doesn't recognize the plant molecule. So instead of delivering a quick read receipt and vanishing, THC lingers on the receptor.
Oh, wow. So it just stays locked in.
It binds and stays there, causing a prolonged, unnatural firing pattern in the neurons.
And what does that prolonged firing actually do to the brain's behavior?
Well, it creates a cascade of behavioral shifts. because CB1 receptors are heavily concentrated in the hippocampus, this lingering activation severely alters memory consolidation.
Which explains why short-term memory is often impaired under the influence of THC.
Exactly. Furthermore, it deeply impacts a vital biological process called synaptic pruning.
Synaptic pruning. We've touched on this before. This is how the brain cleans house while we sleep, right?
You've got it.
Yeah.
Every day, your brain forms thousands of weak, unnecessary synaptic connections based on just random stimuli.
Like seeing a license plate or something.
Right. And during deep sleep, the brain acts like a gardener, pruning away those weak connections so the strong, important neural pathways can thrive.
And the endocannabinoids control this.
Endocannabinoids heavily govern this pruning process. So when THC floods the system and lingers on the receptors, it disrupts the gardener. The pruning process becomes erratic.
Which explains the behavioral hangover.
Yeah.
I mean, if your brain doesn't clean house at night, you wake up with that terrible brain fog, your focus drops.
Your reaction time slows down.
Exactly. And your creativity decreases because your neural pathways are essentially cluttered with biological junk.
That is the exact behavioral consequence of hijacking the CB1 receptor with a highly potent partial agonist.
Okay, so that is THC actively turning the lock.
Mm.
But then there is CBD, cannabidiol.
Yeah.
Our sources note time and again that CBD is non-psychoactive.
Right, it doesn't get you high.
But if it isn't triggering the lock, how does it interact with the receptors at all?
CBD is an incredibly fascinating molecule. It has a very low binding affinity for the orthosteric binding site of CB1.
The orthosteric site meaning the main keyhole.
Right. It doesn't fit neatly into the main keyhole where THC and anandamide go. Instead, it acts as what is known as a negative allosteric modulator of the CB1 receptor.
I'm going to need an analogy for a negative allosteric modulator because that's a mouthful.
Okay, think of the CD1 receptor as a heavy physical door.
Okay.
THC opens the door by walking up, grabbing the handle, and turning it. CBD doesn't even touch the handle. Instead, CBD slides a wooden wedge under the back hinge of the door.
Oh.
Yeah. By binding to a completely different secondary site on the receptor, CBD actually changes the three-dimensional physical shape of the entire protein structure.
So by physically changing the shape of the door, it makes the handle harder to turn.
Exactly. It makes it significantly harder for THC or even our own endocannabinoids to bind to the primary site. The affinity is reduced.
That is brilliant. It actively counteracts the THC.
It acts as a biological buffer. It tempers the psychoactive effects. It dials back the intense anxiety or paranoia that high, lingering doses of THC can often trigger in the amygdala. And at the exact same time it is wedging the door on the CB1 receptor, CBD is incredibly busy binding to all those non-cannabinoid receptors we mentioned earlier.
Like the serotonin ones.
Exactly. It is actively targeting the 5-HT1A serotonin receptors, which is the primary mechanism behind its potent anxiolytic or anxiety-reducing properties.
So THC is aggressively turning the handle, and CBD is wedging the hinge.
That's a great way to summarize it.
But the literature makes it very clear that these two are not acting alone on an empty stage. There are over 100 minor cannabinoids identified in the plant.
Oh, absolutely.
Let's look at the origins of those. Where do these chemicals even come from? Let's talk about CDG, cannabidiol.
Right. CBG is arguably the most important compound from a botanical chemistry standpoint because it is basically the mother of them all.
Wait, what do you mean the mother?
Well, the cannabis plant, while it is actually growing in the soil, does not actually produce THC or CBD directly.
Wait, really? The plant doesn't make THC?
It does not. The plant synthesizes a precursor molecule, an acidic form called CBGA cannabigerolic acid.
Where does it make this?
Deep inside the glandular trichomes, the little frosty crystals on the surface of the plant. Think of CBGA as the raw stem cell of the cannabinoid world.
Okay, so how does it turn into THC then?
As the plant matures, specific localized enzymes within the plant tissue, like THCA synthase or CBDA synthase, grab that CBGA stem cell and convert it into THCA or CBDA.
What does the A stand for? Are these acidic forms biologically different?
The A stands for acid, and yes, the pharmacology is completely different. Because of that extra carboxyl ring on the molecular structure, THCA does not fit into the CB1 receptor.
So it can't unlock the door.
Right, it cannot cross the blood-brain barrier in the same way. If you were to eat raw, freshly harvested cannabis, you would not experience any psychoactive high whatsoever.
Because the keys still have the plastic mold stuck to them. They physically can't fit in the lock.
Exactly.
So how do they become active?
To become the active neutral cannabinoids like THC or CBD, Those acidic precursors must undergo a chemical process called decarboxylation,
Which is the scientific term for setting it on fire.
or putting it in an oven. Applying heat, whether through a lighter, a vaporizer, or, the baking process when someone makes an edible, forces a chemical reaction.
It breaks off the plastic mold.
It breaks off that carboxyl group, releasing carbon dioxide and physically converting the heavy THCA molecule into the lighter active THC molecule. And now it can bind perfectly to the CB1 receptor.
Okay, so CBG is the mother's stem cell. What about CBC, cannabichromene? What does that do?
CBC is a minor cannabinoid that is gaining massive attention in pharmacological reviews. The research shows it is highly beneficial, specifically because of its relationship with our endocannabinoids.
How so?
CBC appears to inhibit the cellular uptake of anandamide.
Wait, so it stops the body from cleaning up the bliss molecule?
Exactly. By preventing the body from pulling anandamide back into the cell to be destroyed by enzymes, CBC leaves more of our natural bliss molecule floating in the synapses for longer periods.
So it naturally elevates the baseline of our own internal thermostat.
Yeah. And several studies in the MDPI stack also noted its potent anti-tumor properties, specifically inhibiting the migration of certain cancer cells, which we will definitely explore later.
Okay, and then there is CBN, cannabinol. And I want to pause here because as the proxy for the listener, I kind of want to push back aggressively on this one.
All right, let's hear.
If you walk into any dispensary today or look at commercial hemp products online, CBN is strictly marketed as the ultimate sleep aid.
Oh, everywhere.
They put like moons and stars on the bottom. They literally call it the sleepy cannabinoid found in old weed.
Right.
But looking at the actual clinical data in our stack,
Yeah.
is that biologically true or is that entirely fabricated marketing?
I am so glad you brought this up, because this is a perfect example of commercial marketing severely outpacing the rigorous science.
That's not true.
Let's look at the facts. It is true that CBN is found in old weed. CBN is not synthesized by the plant enzymes at all. It is strictly a degradation product.
Meaning it breaks down over time.
Yeah, when active THC is exposed to ultraviolet light and oxygen over long periods of time, the molecule degrades and oxidizes into CBN.
Okay, so the old weed part is true. But does it physically make a human being fall asleep?
If we look closely at the exhaustive pharmacological reviews from Frontiers, the laboratory results are highly equivocal.
Equivocal, so basically maybe.
Barely even a maybe. The entire reputation of CBN as a sedative stems from a handful of isolated studies done in the 1970s.
Just a few studies.
Yeah. One study showed that when researchers gave mice heavy barbiturates, injecting CBN increased the duration of the barbiturate-induced
Really?
sleep.
They were already on heavy tranquilizers.
Right. And another study, run right around the exact same time, showed that CBN administered by itself had absolutely no effect on sleep latency or duration whatsoever.
So mice pumped full of heavy tranquilizers slept longer when given CBN. That is hardly a compelling argument for a human sleep gummy.
Exactly.
Why does the reputation persist then?
Well, the clinical reality seems to be that when CBN is administered alongside THC, the combination produces a greater subjective feeling of sedation than either cannabinoid alone.
Oh, so it's a team effort.
But in a recent comprehensive review of human clinical trials, researchers found a complete lack of rigorous clinical evidence supporting a definitive sleep-promoting effect for CBN as an isolated compound.
The human data just isn't there.
It simply isn't there. It is a classic case of anecdotal synergy jumping way ahead of clinical proof.
Which honestly provides an absolutely perfect conceptual bridge here.
Right.
You just noted that CBN only seems to cause deep sedation when it is administered alongside THC.
Right.
A single cannabinoid in isolation rarely tells the whole story. The true biological magic and honestly the source of immense scientific controversy lies in how these hundreds of compounds interact with one another simultaneously. Let's talk about the entourage effect, the symphony versus the solo.
This concept is basically the beating heart of botanical pharmacology, and it is perhaps the most fiercely debated concept in medical science today.
Let's lay the historical groundwork first. The term entourage effect wasn't coined by a dispensary marketer.
No, definitely not.
It was first introduced in the peer-reviewed literature in 1998 by researchers Shimon Ben Shabbat and Raphael Meshulam. What were they originally observing?
What's crucial to understand is that they weren't even studying the plant when they coined the term.
They weren't.
No, they were studying human endocannabinoids. They were looking at the activity of 2-AG, our natural internal messenger.
Okay.
They discovered that the biological activity and receptor binding of 2-AG was significantly enhanced when it was administered alongside two related but completely inactive metabolites.
Wait, inactive? Like they did nothing?
Right. These inactive fatty acid glycerol esters did absolutely nothing on their own. They couldn't even bind to the receptors. But when they were present in the biological soup, they massively boosted the biological efficacy of the 2-AG.
Wow.
Yeah. Mishulam and Ben Shabbat mathematically define this biological phenomenon as a strict inequality. 1 plus 0 is greater than 1.
I love that mathematical expression. 1 plus 0 is greater than 1. The entirely inactive compound amplifies the active one, creating a whole that is greater than the sum of its parts.
Exactly. And over the last 2 decades, this internal biological concept has been applied outward to the cannabis plant itself.
The prevailing theory being what, exactly?
The theory is that a full-spectrum extract of the plant, a botanical soup containing multiple cannabinoids, volatile terpenes, and flavonoids, produces a stronger, safer, and vastly more nuanced therapeutic effect than just taking isolated, highly purified THC or CBD acting entirely alone.
Now, our sources make an incredibly vital distinction here that almost everyone in the commercial space completely misses. There isn't just one entourage effect, is there?
No, there are actually 2 distinct types of chemical interactions happening simultaneously. Intra-entourage interactions and inter-entourage interactions.
Walk us through the difference.
It is all about how the chemical classes talk to each other. Let's start with intra-entourage.
Okay, intra.
The prefix intra means within. This refers to biological interactions occurring between compounds of the exact same chemical class.
So like a cannabinoid interacting with another cannabinoid.
Right. The perfect example is what we discussed earlier. CBD wedging the door hinge to modulate the psychoactive effects of THC. That is an intra-entourage interaction.
Or a terpene interacting with another terpene.
Yeah, exactly.
Got it. And inter-entourage.
Inter-entourage is the cross-class interaction. This is when a cannabinoid interacts with a completely different botanical class, like a terpene or a flavonoid.
Okay, that makes sense.
And this is where the true chaotic complexity of the botanical extract comes into play, because these different molecules hit completely different physiological targets across the body.
So to illustrate just how powerful and exact these interactions have to be, let's look at that deeply specific oncology study cited in the MDPI review.
The Chotel one.
Yeah. They utilized something called the Chotel method. They were testing the synergistic effects of CBD and THC in a petri dish against A2780 human ovarian cancer cells.
The Chotel method is brilliant. It is a highly rigorous mathematical model used constantly in drug discovery.
What does it actually do?
It allows pharmacologists to calculate a combination index, or a CI value. This number definitively proves whether a drug combination is synergistic, purely additive, or antagonistic.
How does the index work? Like, what are the numbers?
It revolves around the number 1. If the CI value is exactly 1, the interaction is purely additive, meaning 1 plus 1 equals 2.
Okay, standard addition.
If the CI value is significantly less than 1, the combination is synergistic. The drugs are actively helping each other, so 1 plus 1 equals 5.
Wow.
But if the CI value is greater than 1, the combination is antagonistic. The drugs are actively fighting each other, canceling out the therapeutic benefit.
So what exactly happened to the cancer cells when they mixed the CBD and THC?
This is where the data gets breathtaking. The researchers took the cancer cells and applied a combination of CBD and THC in a perfect 1-to-1 ratio.
Yeah.
The resulting CI value plummeted to 0.5.
0.5, so massive synergy.
Massive synergy. That 1-to-1 combination was vastly more effective at inducing cell death in the ovarian cancer cells than administering either compound alone, even at much higher individual doses.
We were working together perfectly.
Right. They shifted the ratio slightly to 1-to-2, and it still showed incredibly strong synergy.
But, and this is the absolute kicker that we mentioned at the start of the deep dive, what happened when they shifted the mathematical ratio to 1 part CBD to 4 parts THC?
The entire biological environment flipped. It became fiercely antagonistic.
Wasn't the mark?
The CI value shot all the way up to 6.9.
6.9. That's huge.
Right. At that highly specific 1-to-4 ratio, the CBD and THC stopped helping each other and began actively hindering each other's cellular mechanisms. It drastically reduced the overall anti-tumor effect.
So what does this actually mean for the patient? It proves that botanical synergy isn't just about throwing a bunch of random compounds into a blender and assuming it will heal you, right?
Exactly. Exact mathematical ratios dictate the biological outcome at a cellular level.
So whole plants is better isn't always true.
It's a massive oversimplification.
Yeah.
Yes, the whole plant holds the necessary ingredients, but it depends entirely on the exact chemical profile and the highly specific ratios of the active agents.
Because a slight imbalance can turn a potent medicine into an antagonistic failure.
Precisely.
I really like to visualize the entourage effect, like a massive theatrical production.
Oh, I like this analogy.
Think about it. THC and CBD are your lead actors. They have the most lines in the script, their faces are on the poster, and they demand the most attention. But a play is absolutely nothing without the supporting cast. The lighting crew, the sound engineers, the director.
If the spotlight is pointed in the wrong direction, the lead actor's performance is completely ruined.
Exactly. And in this botanical play, the lighting and sound crew are the terpenes and flavonoids.
That is a phenomenal way to visualize inter-entourage interactions. The terpenes aren't necessarily the ones delivering the heavy dialogue at the CD1 receptor.
You aren't turning the lock.
Right. They are working behind the scenes, they are setting the stage, opening biological doors, altering the mood of the production, and basically ensuring the lead actors can hit their marks perfectly.
So if these terpenes are the backstage crew physically shaping the psychological and behavioral effects of the show, I think it is time we met them.
Let's do it.
Let's look at how microscopic aromatic oils actually dictate whether a patient feels anxious or relaxed, focused, or sleepy.
Well, let's define what we're talking about first. Terpenes are the organic compounds that give cannabis, and honestly, all plants in nature, their distinct pungent smells.
Like pine needles.
Exactly. If you walk through a forest and smell crushed pine needles, or you zest a lemon peel, or you crush a lavender flower in your hand, the volatile aromatic oils hitting your olfactory receptors are terpenes.
And the chemical literature categorizes them heavily by their carbon structure, right? Like monoterpenes versus sesquiterpenes.
Yes. It is based on the number of isoprene units in their molecular chain.
What's the difference?
Monoterpenes have 2 isoprene units, making them lighter and highly volatile. They evaporate quickly. Sesquiterpenes have 3 units, making them heavier, more stable, and more deeply aromatic.
But structurally, what is vital to understand is that pharmacologically, these oils act as incredibly powerful bioenhancers, don't they?
They absolutely do.
What does a bioenhancer actually do? Does it change how the body absorbs other drugs?
Precisely. Let's look at the blood-brain barrier. It is a highly selective, tightly woven cellular fortress designed to keep toxins out of the brain.
Okay, so it's a shield.
Right. Well, some terpenes are known to increase the permeability of that barrier. They act almost like a molecular battering ram.
A battering ram.
Yeah. They temporarily loosen the tight junctions between the cells. This allows larger cannabinoid molecules like THC to cross over from the bloodstream into the brain tissue much more rapidly and efficiently.
Oh, wow. So this profoundly changes the pharmacokinetic profile of the drug.
It hits faster and harder simply because the terpenes open the gates.
That is wild.
Yeah.
Let's highlight some of the major players in this backstage crew then. Let's start with arguably the most prominent one, myrcene.
Myrcene is a monoterpene, and it is one of the most abundant terpenes found in modern commercial cannabis genetics.
What does it smell like?
If you smell a strain that has a deeply earthy, musky, almost clove-like scent, you are smelling myrcene.
And behaviorally, what does it do?
It is heavily associated with profound physical relaxation and heavy sedation. Preclinical studies indicate that local application of myrcene can significantly ease chronic arthritis pain and reduce local inflammation.
Wait, how? Is it hitting the ECS?
Actually, yes, it works through a mechanism that is mediated by the body's cannabinoid receptors, meaning it is directly interacting with the ECS to provide that relief.
And there is a brilliant chemistry detail in the sources regarding myrcene. When myrcene oxidizes, meaning when the harvested plant is exposed to light and air over a long curing process, the molecule structurally changes, doesn't it?
Yes. It changes into a compound called hashishine.
Hashishine.
Yeah, hashishine is a completely unique rearrangement of the myrcene molecule, and it is the exact compound responsible for giving traditional aged hashish its uniquely heavy, rich aroma.
That's super interesting. Okay, next on the call sheet, Linalool, you mentioned crushing a lavender flower earlier. Linalool is the dominant terpene responsible for the scent of lavender, right?
It is, and discussing linalool brings us squarely into the psychological impacts of the entourage effect.
How so?
Linalool has profound anxiolytic, anxiety-reducing, and sedative properties. We have known this anecdotally in traditional medicine for centuries. People put lavender sachets under their pillows to help them sleep.
Right, but now molecular biology allows us to understand the exact mechanism of why it works.
Exactly.
So how does smelling a flower physically alter the wiring in the brain?
Linalool acts as a competitive antagonist of the excitatory neurotransmitter glutamate. Specifically, it binds to the NMDA receptors in the brain.
Okay, let's break that neurobiology down for the listener. What is glutamate, and what happens when it hits an NMDA receptor?
Think of your brain like a car. Glutamate is the biological gas pedal.
Okay, the gas pedal.
It is an excitatory neurotransmitter. Its primary job is to make neurons fire rapidly. When you are under immense psychological stress, or you are suffering from a severe lack of deep REM sleep, your brain essentially gets flooded with glutamate.
So the gas pedal gets stuck to the floor.
Exactly. Your brain is bathed in excitatory signals.
And behaviorally, this leads directly to that terrible feeling of racing thoughts, crippling anxiety.
And the resulting brain fog. Simply because the brain is over-revving and burning itself out.
You lose your ability to focus, your reaction times just plummet, and you feel exhausted but wired.
Right. Your brain desperately needs to enter REM sleep to facilitate an emotional reset, but the glutamate won't let the engine idle.
And this is exactly where linalool steps in.
Linalool acts as a physical block under the gas pedal. It travels into the brain and binds directly to the NMDA receptor.
So it locks out the glutamate.
It competitively locks it out, preventing it from attaching and sending the signal. By blocking that excitatory neurotransmitter, linalool physically dials down the rampant neuronal firing.
Wow. So it promotes deep, profound psychological relaxation.
It helps reset the emotional baseline and facilitates the entry into the deep REM sleep required to clear out the biological junk and lift the brain fog.
That is genuinely incredible. A microscopic, fragrant plant oil is physically blocking an anxiety-inducing neurotransmitter to put you to sleep.
It's not magic, it's mechanical engineering.
It really is. Okay, what about the pines? Alpha-pine and beta-pine.
As the name suggests, they smell sharply of pine needles. Behaviorally, strains high in pine are considered highly uplifting, promoting alertness and memory retention.
But biologically, the deep dive gets really fascinating here. A major 2024 study by researchers Laws and Smid revealed some novel neuroprotective effects, right?
They did. They found that alpha and beta pine exhibited potent anti-aggregatory effects against beta amyloid toxicity.
Wait, beta amyloid. We've covered that in previous deep dives. That is the exact toxic plaque buildup associated with Alzheimer's disease and severe cognitive decline.
That is the one. The pines seem to offer highly specific neuroprotection by inhibiting lipid peroxidation.
So they act as an antioxidant shield.
Yes, protecting the delicate lipid membranes of the neurons from oxidative stress and toxic degradation.
That is huge. And then there is beta-caryophyllene, which is known for its sharp, spicy black pepper aroma. And looking at the pharmacology, this specific terpene completely breaks the rules, doesn't it?
It shatters the rules of botanical classification. By definition, most terpenes do not bind directly to the primary cannabinoid receptors. They hit secondary targets like the NMDA receptor, like linalool did.
But beta-caryophyllene is unique.
Very. Due to its larger, bulkier sesquiterpene structure, it actually acts as a full direct agonist at the CB2 receptor.
The immune system receptor.
All right, so even though it is a terpene by structure, it behaves pharmacologically like a dietary cannabinoid.
By locking directly into the CB2 receptor, it provides highly potent anti-inflammatory and pain-relieving effects.
Oh, yeah.
And it does this without any psychoactivity.
Zero psychoactivity. And there is a behavioral detail buried in the sources about beta-caryophyllene that is just completely wild.
Oh, I saw this in the notes. It involves wild giant pandas.
Yes.
Oh.
Have you ever wondered why wild giant pandas are rolling around in horse manure?
Honestly, no, but now I need to know. Why are they rolling in horse manure?
Because horse manure is packed with undigested plant matter rich in beta-caryophyllene and caryophyllene oxide.
Wait, really?
Yeah. In the freezing winter months, wild pandas will actually seek out fresh horse manure and aggressively rub it all over their fur to coat themselves in the terpenes.
Yeah.
Researchers discovered that activating the CB2 receptor and a specific thermal channel called TRPM8 with beta-caryophyllene remarkably improves the panda's cold tolerance.
You are kidding.
No. The terpene physically changes how their peripheral nervous system perceives freezing temperatures. It literally acts as a biological winter coat.
Nature is utterly bizarre. Okay, before we leave the supporting cast entirely, we have mentioned the flavonoids, specifically a unique class called the canflavins.
Right. Flavonoids are polyphenolic compounds found across the plant kingdom. They are responsible for the vibrant colors of fruits and flowers, the taste of the plant, and they hold massive therapeutic potential.
But the cannabis plant produces specific ones, right?
Yeah, highly complex flavonoids found absolutely nowhere else in nature. They are aptly named canflavin A, canflavin B, and canflavin C.
And what role do they play in the entourage effect?
They show incredibly profound anti-inflammatory and neuroprotective effects. They are likely a massive, understudied component of the behavioral outcomes of cannabis use.
So they contribute heavily to the botanical synergy.
Exactly, by modulating how inflammatory pathways, specifically the COX and LOX pathways, are processed in the body. This provides systemic relief that helps lower the overall stress baseline.
Okay, I have to step in and push back here for a second, because a skeptical listener is undoubtedly thinking something right now.
Okay, lay it on me.
Look, if I can get Lenovol from just sniffing a lavender candle, and I can get pine from walking through a pine forest, and I can get beta-caryophyllene by grinding black pepper onto my steak, why does it matter that they are present in cannabis?
That is a very fair question.
Right. Do these aromatic oils actually change how the cannabinoids themselves behave inside my body?
That is the million-dollar question regarding the interentourage effect, and the answer is a definitive yes.
No.
The mechanism lies in a combination of pharmacodynamic and pharmacokinetic synergism.
Okay, we briefly touched on pharmacokinetics with the blood-brain barrier. Break those two terms down.
Let's start with pharmacokinetics. Pharmacokinetics is, simply put, what the body does to the drug. It is how your biology absorbs, distributes, metabolizes, and excretes a compound. As we discuss, terpenes like myrcene act as battering rams, altering the permeability of the blood-brain barrier.
So they actively improve the absorption.
They actively improve the absorption and tissue distribution of the cannabinoids. They ensure that a higher concentration of the active THC or CBD actually reaches the target receptors in the brain rather than being filtered out by the liver.
Got it. And pharmacodynamics.
Pharmacodynamics is what the drug does to the body. It is the study of the biological and physiological effects at the receptor level.
So hitting the actual locks.
Think about the complexity we just uncovered. Even if a terpene doesn't bind to the CB1 receptor like THC does, it might be binding to the NMDA receptor to block glutamate, like linalool.
Right. Or it might be hitting the 5-HT1A serotonin receptor to reduce anxiety.
Exactly. By hitting multiple cellular targets simultaneously across completely different physiological networks in the brain and immune system, the combined botanical extract can disrupt complex cellular resistance mechanisms.
So it's a multi-pronged attack.
It produces a comprehensive, multi-layered behavioral shift that a single, isolated cannabinoid molecule simply cannot achieve on its own.
Wow. Okay, but listening to this staggering level of complexity, I mean, we are talking about hundreds of cannabinoids, volatile terpenes, and unique flavonoids, all interacting in highly specific mathematical ratios that can flip from synergistic to antagonistic in an instant.
It's a chaotic botanical soup.
Yeah. Standardizing this for a medical patient seems utterly impossible. How does the medical world even attempt to categorize this? Let's look at the real-life behavioral impact of chemovirus and symptom management.
Well, the very first step to standardizing this medicine is that we have to completely throw out the old culturally entrenched terminology.
Like sativa and indica.
Exactly. If a patient walks into a typical commercial dispensary today, almost every single product is heavily labeled in a binary system, sativa or indica.
Right, sativa is relentlessly marketed as the uplifting, energizing, creative daytime medicine.
And indica is marketed as the heavy, sedating, pain-relieving nighttime medicine.
The old joke being indica means into couch.
Right. But scientifically, chemically, and genetically, those labels are completely and utterly obsolete.
Really? Why? Where did they even come from?
They were originally botanical classifications from the 18th century that referred strictly to the physical morphology, the shape of the plant.
The shape of the leaves.
Yeah. A sativa plant grew tall with narrow, thin leaves. An indica plant grew short, bushy, with broad, wide leaves.
Okay, but that doesn't mean anything chemically.
Fast forward to today, after decades of rampant, undocumented crossbreeding in the underground market, almost everything in existence is a massive genetic polyhybrid.
They're all mixed up.
The physical shape of the leaf tells you absolutely nothing about the chemical ratios produced in the trichomes. A tall, thin plant could easily produce a massive amount of sedating myrcene.
So if indica and sativa are biologically meaningless, what does the rigorous scientific community use to categorize the medicine?
They rely entirely on chemovars, which is short for chemical varieties.
Okay, chemovars.
It is a classification system based strictly on the tested, laboratory-verified chemical profile of the harvested plant, regardless of what the plant looked like while it was growing.
And the European Pharmacopoeia has actually stepped in and standardized this into 3 main overarching types, haven't they?
Yes, to provide a baseline for clinical trials, type I chemovars are THC dominant.
Okay.
Type 2 chemovars have a highly balanced, roughly 1-to-1 ratio of THC and CBD. And type 3 chemovars are CBD dominant with trace or near zero amounts of THC.
But as we've learned from the Châtelet math, just knowing the macro THC and CBD ratio isn't nearly enough, is it? The backstage crew, the terpenes matter just as much.
They dictate everything.
So how does this complex chemovar selection actually play out in real-life symptom management for a patient?
Let's look at a deeply revealing clinical study by Vigil and colleagues, which was heavily referenced in the MDPI Pharmacological Review.
What do they look at?
They analyzed how highly specific chemovars affected a large cohort of patients dealing with overlapping symptoms of chronic pain, severe anxiety, and clinical depression.
A lot of interconnected symptoms there.
Very interconnected. They tracked the data and found that minute, subtle shifts in the ratios of myrcene, caryophyllene, THC, and CBD drastically changed the behavioral outcomes for the patients.
Give me a specific example of how a slight shift altered the outcome.
Take a specific chemovar they tested, labeled MC61. It had a highly potent type I profile high THC, around 20 to 25%.
High THC.
It had very low CBD and specific low amounts of myrcene and caryophyllene. The data showed that while this specific profile was highly effective at agonizing the CD1 receptors to distract the brain and treat the physical pain,
It killed the pain bud.
it had a disastrous side effect. The lack of buffering CBD and the lack of anxiolytic terpenes meant the high THC levels inadvertently exacerbated the symptoms of anxiety and depression in the patients.
Wow. The cure for the pain became the catalyst for the panic attack.
Exactly, and this connects deeply to the behavioral reality of chronic illness. A chronic disease is rarely just one isolated symptom. It is a vicious, compounding cycle.
Right. Unrelenting chronic pain leads directly to poor, fragmented sleep.
And that severe lack of sleep floods the brain with glutamate, leading to crippling daytime anxiety.
And that anxiety overworks the brain, leading to profound brain fog. Because of the brain fog, the patient's focus drops, their reaction time slows, their creativity vanishes.
And their overall quality of life just plummets. They are trapped in survival mode.
So effectively treating the patient isn't just about throwing a massive dose of THC at the pain receptors.
Not at all. It requires incredible precision.
So what's the alternative?
By selecting the correct, highly specific chemovar, perhaps shifting the patient to a type 2 profile with a much higher CBD content.
To chemically wedge the CB1 receptor and buffer the THC-induced anxiety.
Right, and combined with a high concentration of lindyl to physically block the glutamate at the NMDA receptors.
So a doctor doesn't just treat the physical pain, they intervene in the cycle itself.
They reset the patient's psychological baseline. The linalool and CBD allow the patient to actually achieve deep REM sleep.
And the sleep clears the neurotoxic brain fog. They wake up refreshed, their appetite is restored, perhaps aided by CBG or a low dose of THC, and their focus and behavioral functionality return.
Okay, but here is a massive, incredibly frustrating real-world pushback.
I think I know where you're going with this.
If the pharmacology is this precise, if getting the wrong chemovar can literally trigger a panic attack instead of relieving pain, Why are severely ill patients so often left to just guess at a commercial dispensary?
It's a huge problem.
Why aren't highly trained doctors prescribing exact chemovars with calculated terpene ratios?
It represents a critical systemic failure in the current medical education system. There is a massive gaping hole in medical training regarding the endocannabinoid system.
Just a lack of education. The sources we reviewed included a highly illuminating statement and survey from the Beth Israel Deaconess Medical Center, led by Dr. Kian Taranchi.
What did the survey say?
They surveyed oncology fellows doctors training to treat the most complex cancers and found that these highly educated physicians are woefully, dangerously unprepared to answer basic clinical questions about cannabis.
Why? Is it the stigma?
Mostly, it is simply not taught in the standard medical school curriculum. The vast majority of doctors cannot confidently advise a patient on specific chemovars simply because they don't know what a chemovar is.
That's terrifying.
And they are uneducated on severe potential drug interactions. For example, CBD acts as a potent competitive inhibitor of the cytochrome P450 enzyme system in the liver.
Wait, what does the cytochrome P450 system do?
It is the liver pathway responsible for metabolizing over 60% of all standard pharmaceutical drugs.
Oh, wow. So a patient takes a massive dose of CBD.
It occupies those liver enzymes. If they then take their standard heart medication or a chemotherapy drug, the liver can't break it down.
Because the enzymes are busy with the CBD. So the pharmaceutical builds up to highly toxic levels in the bloodstream.
Exactly. And because doctors aren't trained on this, vulnerable patients are left to self-medicate, relying entirely on the anecdotal advice of a 22-year-old dispensary clerk with zero medical training, rather than receiving rigorous clinical guidance.
Which is truly terrifying when you are dealing with severely immunocompromised individuals or complex chemotherapy regimens.
Absolutely terrifying.
But despite this glaring lack of formal training, the sheer biological efficacy of these botanical compounds is violently forcing mainstream medicine to pay attention.
It really is. It is actively pushing cannabinoid science out of the commercial dispensary and into the sterile palliative care ward.
Which brings us to the ultimate proving ground for this chemistry: frontline biology. We are talking about oncology, chemotherapy side effects, and severe neuropathy.
This is where the theoretical biology meets life or death clinical application. We are talking about utilizing these molecules to help patients undergoing the most physically and psychologically grueling medical treatments in existence.
Let's start with behavioral side effects of chemotherapy. The physical toll of chemo is devastating.
Severe loss of appetite, relentless nausea, violent vomiting, massive sleep disturbances, and a crippling systemic fatigue.
How is the medical community utilizing cannabinoids to combat this?
This is actually one of the most well-established, legally recognized areas of cannabinoid medicine.
Because the mechanism works.
Because the mechanisms are so undeniable, synthetic single-molecule cannabinoids have been federally approved and prescribed for decades.
Like what?
Pharmaceutical drugs like dronabinol, which is essentially a synthetic version of pure THC, and neboloni. They are actively prescribed to stimulate appetite in cachexia patients and to drastically reduce chemotherapy-induced nausea and vomiting.
So they are actively using the THC mechanism. They are sending that key into the CB1 receptors to trigger the hunger signals in the hypothalamus.
And simultaneously binding to the receptors in the brainstem to quiet the nausea reflex.
Yes. What about nerve pain? Severe peripheral neuropathy is a massive, debilitating side effect of several prominent chemotherapy drugs.
It is, specifically with taxane-based chemotherapy drugs like paclitaxel.
What does paclitaxel do?
Paclitaxel causes massive damage to the peripheral nervous system, resulting in severe neuropathy, which manifests as agonizing burning, tingling, and sharp electrical pain in the hands and feet.
It can be so debilitating that patients have to halt their life-saving cancer treatments, right?
Exactly. But the pharmacological reviews highlight multiple studies demonstrating that CBD is remarkably effective at alleviating paclitaxel-induced neuropathic pain.
But how does CBD physically do that? We already established that CBD does not bind to the main CB1 pain receptors in the brain. It wedges the hinge.
Right.
So how does it stop nerve pain in the hands and feet?
Remember that serotonin receptor we unpacked earlier? The 5-HT1A receptor?
Yes, the mood and anxiety one.
In rigorous in vitro studies utilizing breast cancer models, researchers identified that the direct activation of this highly specific serotonergic pathway is the exact mechanism through which CBD exerts its analgesic effect.
So it's hitting the serotonin receptors instead of the cannabinoid ones.
Yes. CBD travels to the 5-HT1A receptor and modulates the pain perception signals as they travel from the damaged peripheral nerves back up the spinal cord to the brain.
It intercepts the pain signal before the brain can fully register the agony.
Precisely.
Okay, so we know definitively that the endocannabinoid system can be leveraged to manage the horrific side effects of cancer treatment.
We do.
But what about the cancer itself? Can cannabis compounds actually shrink tumors? And I want to step in here and be incredibly careful, because as we established in our mission statement, we must objectively report the data from the literature without picking sides, without endorsing unproven cures, and without feeding into the internet hype.
I agree completely. It is absolutely vital that we remain deeply impartial and rigorous here, because the clinical data regarding tumor suppression is incredibly complex, highly nuanced, and in some cases, wildly conflicting.
Let's lay out the conflicting data directly from the sources. On one side of the coin, we have peer-reviewed studies showing aggressive tumor suppression.
Yes, we have significant preclinical data demonstrating that specific cannabinoids can actively induce apoptosis in tumor cells.
Apoptosis meaning programmed cell death.
Yes, normal, healthy cells have a built-in self-destruct sequence when they become damaged. Cancer cells mutate and turn off that self-destruct sequence, allowing them to multiply uncontrollably.
And cannabinoids fix that.
Cannabinoids have been shown in specific cellular models to turn the self-destruct sequence back on, forcing the cancer cell to dismantle itself.
Wow.
Furthermore, they can inhibit angiogenesis, which is the process by which tumors build new blood vessels to feed themselves.
By cutting off the blood supply, the tumor starves.
In fact, this mechanism is so promising that a drug called Sativex is currently in clinical trials. Sativex is an oral botanical spray containing a highly standardized full plant extract.
And what is the chemical makeup of Sativex?
It is a precise 1-to-1 ratio of THC and CBD, accompanied by the plant's natural terpenes.
The exact golden ratio from the Chart-Holloway mathematical method we talked about earlier.
Yeah.
The CI of 0.5.
Exactly. Sativex is currently in world-first phase 2 clinical trials being tested directly alongside temozolomide, which is a standard, highly aggressive chemotherapy drug.
What are they testing it on?
They are testing this combination specifically for patients suffering from glioblastoma, which is one of the most aggressive, fast-moving, and deadly brain tumors in existence.
And the results?
The preclinical model strongly suggests that the 1-to-1 botanical extract might significantly enhance the chemotherapy's ability to breach the cancer cell's defenses and kill the tumor by disrupting the EGF and EGFR signaling pathways.
That sounds incredibly promising. But, as we said, the data is conflicting. What does the other side of the coin look like?
On the other side, the pharmacological reviews highlight a highly critical study conducted by researcher McCallop and colleagues.
Okay, what did they find?
They were investigating the biological effects of THC on specifically targeted breast cancer models. And their data revealed a terrifying possibility.
A terrifying possibility.
They found that introducing THC to this specific type of cancer might actually enhance breast cancer growth and actively promote metastasis, which is the spreading of the cancer to other organs.
Wait, stop. It made the cancer grow faster. How is that biologically possible if THC induces apoptosis in a brain tumor?
Because of the CB2 receptor. Remember, THC binds to both CB1 in the brain and CB2 in the immune system.
Right.
Breast cancer is highly dependent on the immune system's response. In McCallop's model, the THC bound heavily to the CB2 receptors on the patient's immune cells.
Oh no. So it shut down the immune response.
By agonizing those receptors, the THC severely suppressed the natural anti-tumor immune response. It essentially handcuffed the white blood cells that were actively trying to fight the tumor, creating an immunosuppressed environment that allowed the cancer to multiply and spread unhindered.
So in a glioblastoma brain tumor model, a 1-to-1 extract actively helps kill the cancer cells.
Yeah.
But in a specific breast cancer model, isolated THC suppresses the immune system and actively helps the cancer spread.
Which is exactly why rigorous, tightly supervised, long-term clinical trials are absolutely necessary, and why self-medicating with dispensary cannabis to treat a severe oncology diagnosis is incredibly dangerous.
You simply cannot assume that cannabis cures cancer as a blanket statement.
You can't. The biological effects are fiercely dependent on the specific type of cancer, the exact mathematical ratio of the cannabinoids, the presence of specific bioenhancing terpenes, and the unique, highly variable immune system of the individual patient.
Let's bring this profound complexity back to the entourage effect for a moment. When evaluating tumor suppression, the sources brought up a really fascinating comparison between using a highly isolated pure compound versus using the whole botanical plant extract.
They called it the shotgun versus the silver bullet approach.
This is perhaps the most paradigm-shifting finding from the breast cancer preclinical models we reviewed.
It really is. When researchers took a pure, highly isolated THC molecule, a single perfected chemical, the silver bullet, and applied it against breast cancer cells in a petri dish, it demonstrated a certain baseline level of anti-tumor activity.
It worked, to a degree.
But then they ran the test again. They utilized a full botanical extract, the shotgun approach.
And what was in the shotgun blast?
This extract contained the exact same milligram amount of THC as the first test, but it was accompanied by the messy, chaotic botanical soup of minor cannabinoids like CBG and CBC, and the highly volatile bioenhancing terpenes.
And what happened?
The full botanical extract was significantly mathematically more potent at inducing cell death in the cancer cells than the isolated silver bullet.
Wow. So the supporting cast, the backstage lighting and sound crew, actually helped the lead actor kill the cancer cell.
Yes. The presence of those minor cannabinoids acting as allosteric modulators, combined with the terpenes altering the cellular permeability, seemingly allowed the botanical extract to attack the malignant cancer signaling network from multiple angles simultaneously.
Because cancer is incredibly adaptive, right? It builds resistance to single drugs very quickly.
Right. But the shotgun approach overwhelms the cell's resistance mechanisms, forcing the apoptosis self-destruct sequence to trigger.
Okay, here is where it gets incredibly profound.
Yeah.
If Sativex, which is essentially a full plant one-to-one shotgun extract of THC, CBD, and terpenes, is currently sitting in world-first clinical trials for highly aggressive brain tumors, what does that mean for the entire future of synthetic single-molecule pharmaceuticals?
Well, if we pull back and connect this singular botanical plant to the broader scope of human medicine, it actively challenges the very foundational philosophy of how we have manufactured drugs for the last century.
Let's pull all this complex biology together to see the big picture. We started by looking inward at our own biological hardware, the endocannabinoid system.
Our master internal thermostat.
Right. Utilizing retrograde signaling and how it deploys anandamide across CB1, CB2, and serotonin receptors to maintain absolute cellular homeostasis.
And we saw how the plant produces phytocannabinoids that act as mimic keys to those exact biological locks, with THC aggressively turning the handle to alter memory and synaptic pruning.
And CBD sliding a wedge under the hinge to fundamentally alter the shape of the receptor.
We explored how the true magic and the overwhelming mathematical complexity lies in the entourage effect.
The intra and inter-entourage chemical interactions where highly volatile terpenes act as the backstage crew, myrcene battering down the blood-brain barrier.
And linalool physically blocking excitatory glutamate to shift a patient's entire psychological and behavioral baseline.
Intervening in the vicious cycle of chronic illness, allowing a patient to achieve deep REM sleep, clearing their neurotoxic brain fog, restoring their focus, and actively managing the devastating behavioral side effects of grueling treatments like chemotherapy.
It is a profound, interconnected cascade. It starts at the microscopic cellular biological level with a lock and a key, bubbles up into the psychological feeling of anxiety relief or deep sedation,
and culminates in the behavioral ability of a suffering patient to actually function, eat, sleep, and create in their daily life.
But as we close this out, I want to leave you, the listener, with a final, slightly provocative thought to mull over regarding that shotgun versus silver bullet analogy.
Go ahead, lay on them.
Modern pharmacology as an institution has spent the last 100 years desperately trying to isolate single active ingredients. They want to find the one molecule they can purify, synthesize, patent, and sell as a targeted silver bullet. Right, like how we isolated aspirin from willow tree bark, or penicillin from a specific mold.
Exactly. It is the absolute, unquestioned bedrock of the modern pharmaceutical industry. But if the entourage effect continues to bear out in clinical trials, if it proves that complex, chaotic botanical synergies, the shotgun approach of hundreds of interacting terpenes, flavonoids, and minor cannabinoids acting simultaneously across a dozen different physiological pathways, are actually required to bypass cellular resistance and heal the human body. Does cannabis fundamentally break the traditional pharmaceutical model of medicine?
That is a massive paradigm-shifting question.
Isn't it?
And the truth is, it is going to take decades of highly rigorous, deeply impartial clinical trials to fully answer it. and hopefully a massive overhaul in medical school training for our frontline doctors so they can guide patients through this complexity safely.
We are just scratching the surface. We are at the very beginning of understanding what this plant can teach us about our own biology. Well, to you, the learner listening right now, thank you for sharing your stack of sources with us and prompting this deep dive.
Yeah, the muddy waters of diagnostic marketing and anecdotal medicine are incredibly difficult to navigate.
But by throwing out the hype, ignoring the dispensary labels, and sticking strictly to the molecular biology, the exact chemistry, and the peer-reviewed clinical data, we can finally start to see the mechanisms clearly. Keep asking the hard questions, keep looking past the marketing to the actual data, and keep learning.